So we have our next speaker is Rui Chen from University of California, Irvine. So thank you very much.
Okay, thanks for the organizer for the invitation. I don't have any financial disclosure. So thanks for the previous speaker, Dr. Kenyon. And he already laid out the basic genetic landscape of the AMD. So I think commonly to say there's clearly an importance of genetics influence on AMD risk. And although we have not been able to use the genetic to guide the therapy, but it's an opportunity for us to really to understand the causal genes and variants, and then so that we can understand better of the genetic diseases, the mechanism of the disease, but also potentially develop therapy and maybe even personal therapy. So, the one of the major way to get to the genetic factors under AMD has been, because it's a complex disease, have been using this sort of GWAS study, genome-wide association study, as Dr. Kenyon already mentioned. But one of the challenges for this sort of study is the follow-up to identify the causal variants on the, highlighted by those studies, because the SNPs or are tagging SNPs, or are basically tag haplotype that connect association between this particular region and haplotype to the disease. And most of the tagging SNP are actually turn out to be not in the coding region, because most of our genome is made of non-coding region. And therefore, to connect that variant to a gene could be very challenging. Not necessarily the gene that nearest to the variant is the causal gene. And also, one of the limitation, just a few of limitation of this sort of study is really looking for germline variant connection or association with the disease risk. So there's no cell context are basically illustrated from that study. We don't know which tissue and cells that matter for it. So turn to this from this sort of association to causality is actually one of the major challenges in the complex genetic field. And what I'm trying to do today is just to share some of my own thoughts on how we can overcome this challenge. So basically, starting from GWAS results, we have the variant and phenotype, right? The variant in this case are this, initially this 37 loci, now it's 60, 70 loci to the phenotype, which is basically AMD. So we need sort of to make this association to causality, we need to fill the gap, essentially. The variant, in this case, because the variant, as I said, 95% of them are non-coding. So there's a lot of potential function of this variant and genes. So it could be, for example, gene regulatory elements, could be splicing, could be RNA stability, could be post-transcription modification, so on and so forth. But today I will just use the gene regulatory elements or variants as an example to illustrate, because the process is sort of similar, but slightly modification, I think. So in the context of the gene regulatory elements or variant, so we need to basically fill in this gap. It's basically you need to map the variant to a transcription regulation elements, and then the element function on gene or genes of its targets. And gene need function in particular cell context. Could be one cell type, could be multiple cell types. And then that cell types together and lead to phenotype, right? So essentially there's a big gap to fill. And that's exactly what the challenge is. So we can sort of break it down in that way. So we need to identify essentially the tags, identify variants that affect the gene regulatory elements. So we, instead of jump from here, we can just jump this first step first. We also need to identify all the gene regulatory elements in the cell context, right? So which element target what gene, right? This is the second gap. And third one is basically link the regular elements to its target gene. And also we need to characterize gene function in a specific cell context. What does gene do? And finally, with those information, then we can hopefully connect the cell to phenotype. So if we can break down these sort of multiple steps and try to fill all those gaps, perhaps we can link this variant to the phenotype at the end. So from the GWAS, what we got is basically variant and gene. There's many methods has been developed to basically try to narrow from the or prioritize the causal, the social variant or genes, potential genes, to the causal ones. So a lot of them are basically, for example, use the statistical fine mapping method, looking for use Bayesian method to identify the highest probability of variant that in this LD block, for example. And there's also a method such as colocalization, basically try to overlapping with the SNPs identified from GWAS to some sort of functional annotation, and then to say, oh, this particular variant in this LD is actually overlap with this functional variant. From the gene side, we can start with from like gene expression, and then the SNP training, and then get the TWAS transcript, genome-wide association to nominate a genes that are more likely to be associated. For example, we can base on gene expression, gene set enrichment method, and pathways to basically prioritize or give a high probability that gene is likely to be involved in the process, and build on the knowledge we know. And also gene burden analysis, for example, looking for, instead of common variants, you can look at rare variants to see what rare variants are more enriched in the patient cohort than the other. And a lot of time we're really using a coding variants, which basically, skip make the mapping from variant to genes much easier. So we can look at the sort of burden test, whether individual rare variants or set of rare variants on gene sets, and then to see, oh, this gene is highly enriched from particular deteriorate variants, then likely to be the right gene, for example. And some functional assay, right? So if we know some of the system, then we can test the gene function. Based on function, we can infer this gene more likely. So those are typically, there's many other things I don't list, but it's sort of the way people are working on. Well, one thing, as I said, if we're looking for gene regular variants, then there's another link we can make. So that's basically, So one thing you can link variant directly to gene is through like the QTL type thing. For example, the expression QTL in this case is looking for variants that's changing the quantitative gene expression in a large data set. And then if a variant has associated with some target gene expression, then that's sort of link. So one thing in this case is we also can, instead try to directly link to here, we can go a mediator, basically the element as I mentioned, because variant function and the gene expression through as part of the gene regular elements. So if you can map the gene regular elements here, then what we can do is here we have a lot of different methods, to identify them. For example, we can look at for the clumping structure, open clumping region for cell or tissue. We can look at histone mark, acetylation, and then lysine methylation, markers. So there's particular signature for enhancers. We can look at the enhancer RNA expression. So many experiment methods along with the prediction method to identify potential gene regular elements. So if we can have those information, then we can do an overlapping between variant to element, and then we can also then connect element to a gene. For example, one of the ways to from high C to looking for which basically topology interaction between enhancer and promoter region, and that will link the enhancer to genes. So if you basically put element in the middle, then you have even more route potentially you can reach from variant to gene, basically. So, and then lastly, if you really want to get this, high valuable, high likely, causal variant, you actually what you do is typically you integrate everything together and try to make the best sense of it, and to say, oh, which one is most likely to be the right variants and gene to test with. So, this is the paper published by Anand Swarup in Nature Genetics. So to see whether the eQTL in this case will be helpful in mapping the potential variant in the GWAS hit. So this is the figure from their paper. So what basically you can see this, what they have done is they did, they call IGTAX. The reason called IGTAX because a large consortium doing GTAX, gene environment interaction, tissue, gene tissue environment interaction. And then they have like 800 samples, but they are all those samples. Those are different parts of the body, but they left out retina. So what Anand Swarup group did is actually collect 453 samples, and then they are staged at different AMD stage, and then they perform the RNA-seq on the retina tissue, and then you can clearly see the retina, if you use PCA analysis, are actually separate in terms of transcription, separate from other tissues, indicating it has its own very unique signal. And then you can see clearly if you do dendrogram, it's closer to brain, which makes sense because they are connected, as we know, and then they are quite separate from many other tissues. So with this information, so I think there's a table back there. So basically what they find 14,000 cis-EQTL in the retina. About a third of them are retina specific, telling you there's a lot of tissue specific EQTL need to be there. And by doing the mapping, what they find from these 37, at the time 37 loci, they find 6 of them, they have nominate, very strong like candidate or leading SNPs that are EQTL overlapping with the GWAS signal, and presumably so-called hypothesis to test the causality. So that's actually great, meaning showing you to look at tissue-specific expression is very powerful in trying to map to the causal variant. But on the other hand, it's only 20% of the loci are mapped this way, right? So it's 80% of them you still don't know, even if we have this quite large data set. So what's missing? So there's some limitation in the study. In a sense, the first way is related to the bulk tissue method they use. Right? You take the retina. We all know there's many different cell types. Maybe the most important cell type, not necessarily the most abundant ones. So the data will be really biased toward this most abundant data. So the tissue will not give you which cell types are important there, even in that EQTL. But also, you don't get cell type specific EQTL if such thing exists, right? So given you have a tissue 30 to 40% EQTL identified retina are retina specific, it's not unreasonable to think cell type, there's a cell type specific EQTL as well. Right? So, and then also, you don't have cell type specific regular elements identified from that study. That study only have RNA-seq, so you don't have the open chromatin information there. And of course, as we know, there's the RP and core is actually probably even more important than retina in terms of GWAS. So you need to expand out to other tissues as well, like RP core in this case. And maybe by overcome those limitations, you'll be able to expand more if still a large fraction of unexplained GWAS is actually due to the gene regulatory elements or gene regulatory changes. So to test that idea, we actually did a quick pilot study where we performed single-cell RNA-seq and single-cell attack-seq, looking for each cell type, the gene expression and the open chromatin profile, and then try to build a EQTL and to see whether there are cell type specific EQTL. This is a very small pilot study. It's only have 20 samples-ish. So, but what we find is what you're looking at is the cell type those are the retina. Again, those are the retina, and then this number of QTL. So what you can see, the yellow ones are basically QTL only specifically observed for that cell type, not across different cell type. So then we worry about it's only 20 and it's really underpowered, and whether what we see is just artifact. So then what we have done is to compare, to overlap those QTLs with other bulk study. So what we find is, this is the blue one, this is the one percentage that overlap with the bulk retina QTL. I just showed you from group study, and then the other color ones are basically EQTL overlap with other type of tissue, bulk tissue observed. So you can see a lot of them are actually confirmed by other study. But nevertheless, this 44%, which is back here, are basically cell type specific and novel EQTL, never being observed. So suggesting that there's very likely there's a large number of EQTL that is sort of missed through this sort of bulk study. So if that's true, then maybe identify those EQTL would be really useful resource to help us to mapping the, for example, the potential causal variants. So the hypothesis we put out and we're thinking about is really the single-cell omics profiling could improve the fine mapping of GWAS SNP. to identify causal variants and the regular elements and genes, and also the relevant cell types, because the intrinsic advantage of this method. So here's our sort of overall design. So if we take the donor tissue, the retina or RPE choroid, and related tissues, we can perform the single-cell RNA-seq, where we're basically looking for transcriptome for individual cell, and then we can cluster them based on their cell type. So basically identify the transcriptome for each cell type in the tissue. We also, in parallel, can perform the single-cell attack-seq, which is looking for open chromatin region for the corresponding cell. And then we can also identify the basic open chromatin each cell type. And then, in addition to that, we can add, because those are from donor, we also perform the whole genome sequencing or genotyping of it. So then from these three data type, what we can do is we can look at, for example, the gene pick link. I will show you a little bit later, to basically looking for open chromatin association with the target gene. We can also looking at open chromatin profile, and then we can also looking at eQTL here in this case, because we have genetics here. We look at, for each cell type, look at the single cell eQTL. And then in addition, depends on the data type, we can also look at allele-specific expression, and then the allele-specific open chromatin accessibility, so on and so forth. So for those ones, basically we'll be able to map from the sort of variant to element to gene. And then in there, we can bring into the GWAS loci, try to do colocalization and the fine mapping, so then we can identify, nominate potential causal gene and variants. So that's basically the overall design. So to do that, we need to have a very good category of this basic open chromatin profile and the gene expression, and to cast a wide net to really, how to say, like get a comprehensive cell type and gene expression profile and then chromatin over the course of the aging process. So as part of the Human Cell Atlas, our group assembled this about 100 donors, and they have a wide range of age from 0, basically 1 day of age, to 94. And then they have male, female, and this is a different ancestry background. So we perform single-cell RNA-seq and attack-seq on those. So this is a quick look at UMAP on the retina. In this case, it's about overall. And also we integrate quite a few publications that are already published that in public domain, and then integrate in this map. So together, there's 3.4 million total number single cell, single nuclei, and from 122 donors in this case. And then there's more than 120 cell types identified. Each color highlight the major cell types here. So we did that for the entire part of eye as the single cell atlas project. You can see the ciliary body, TM, limbus, sclera, lens, iris, cornea, optic nerve head, so on and so forth. So each part we have about 1 million nuclei and cell profiled from 50 to 80 different donors, depending on which part. And then together, there's almost more than 350 cell types in the eye identified. So in addition to the single-cell RNA-seq, we also perform single-cell attack-seq on the retina. It's about 400K. And then to your, so this is basically co-embedding. And this is basically, again, a UMAP showing different cell type, different color, different major type of the retina. And then this light color is the attack-seq cells. And then you can see they co-embed nicely with the RNA itself. So from here, we can see all the open chromatin profile for all cell types in the retina. So with this information, do we gain information? And the answer is yes. So from the retina, we identify 670K open chromatin region, and this is the distribution of those open chromatin region across different cell type. And then the amount, so this is 3 times more than the open chromatin region identified from bulk data. So you can see there's a lot of cell type specific, and you can see here there's about 20% of them show actually cell type specific openness. So with this sort of integrated information, what we can do is try to link the open chromatin region to the gene. So this is just schematic where you can separate the open chromatin region into 3 categories. the first criteria is basically open in all cell type. But of course, some of them are sort of stable, do not change at all across different cells, but some are changed. You also have a subset of cells which have the constant peaks and also variable peak, and then you have cell type specific ones. So this is the so what we do is like we're looking basically for correlation between the openness or accessibility of those peaks to either the gene expression of the nearby gene or the the openness of the promoter region of that gene. So by looking at this correlation, we'll be able to sort of make the link, the association. So this is just a breakdown of different type of the peaks. So and then and by linking them, and you can see the variable ones. Actually, about half of them actually have so we also did single cell Cotton tag, which also looking for the histone marks for each peaks. So we can see there's a significant enrichment for the variable peaks to the enhancer activity, active enhancer marks. But you do have around 50% of the peaks, right? This is positive for the enhancer mark actually appears stable or not changed across their cell type. So that underscores that correlation is important, but also looking for just different ways to look at the enhancers is actually also just as important. For example, looking at these histone marks. So, but in any case, about like 25% open chromatin, if based on correlation alone, we can identify their target, or particular target gene. This is basically looking for the target gene ataxic accessibility and to the gene expression. You can see it very nicely correlated, and because that's what we used to make the link. So with this exercise, is that useful? So we start using the sort of this map the chromatin region, and then we start looking at this 37 loci again. So this is the so we do find several loci that we can link now with the retina single cell data. This is just one example, and this is the TGF-beta receptor 1. And then you can see in the retina, what you see the gene expression is really highly expressed, specifically in Macular glial cells, which is about like 10% of the cell population in the retina. And this is the initial GWAS. You can see they have sort of a wide. It could be this gene and this gene and that gene, or even further away. So, but then when you look at the chromatin region, this the consistent ones. This is the promoter. And then if you look at Macular glial cell where the gene expressed, you can see a unique peak here just next to it, which basically corresponding to this sort of intron 1 region of this gene. And then if you look at the core localization, then they highlight this particular SNP within this peak that also can have strong correlation in terms of accessibility to the gene expression. So that actually, we calculated the posterior probability score, and then it's greater than 0.9, and suggesting this particular variant are likely to be causal for this gene, and then for this TGF-beta receptor 1, and then particularly in Macular glial cells. So that's basically the hypothesis. Of course, you need to test functionally, but at least it seems to me that even from our current limit data, you'll be able to map more loci and nominate more loci from this EQTL sort of study. So, in addition to looking for cell type specific, this context, with the increase in a number of cells profile, you actually can even more sort of fine mapping at the cell subtype level. So this is just an example. This is the UMAP of the RP choroid. We put together the reference. The total number of cells is 851K, and then 17 major cell types and 45 subtypes. So if we zoom in this particular plot, the red ones are RPE cells. If we zoom in, it can be further separated into like 5 subclusters. So without going into the detail, and this is Barty's group from the picture from their study, and then they can clearly show the RP is not homogeneous, so they can be separated into based on the morphology, physiology, and function. Potentially, you can separate them into this sort of concentric ring situation. So, because when we did the atlas, we did take this region, RP region, and also the peripheral region, and then that allows us to basically perform sort of trajectory analysis, and so to identify whether these 4 clusters corresponding to different part of the RPE. And then our current conclusion, and then recently also we validated from the single-cell spatial experiment, is that yeah, these 4 clusters corresponding to different part of the RPE. With this, the green one is more at peripheral, and then the yellow one is at the more central. As we know, AMD do not occur uniformly across the whole retina, so it's possible the RPE at different region may have different implication in the disease. So what we have done is we take the GWAS gene list, and then we perform the gene expression enrichment set assay. And this is the AMD. I just want to point to the last row, and you can see the most central subcluster of RPE is the one that show higher expression enrichment for AMD genes than the peripheral ones. So that gave us some interest, lead to maybe you can look specifically looking at this particular RPE cluster and do a more follow-up study. So I think so what I have shown you, I talked about is people when try to get the causal variant gene, most of the methods are really based on either the statistic probability model argument or some correlation with the functional annotation. So I think it would be amiss to not talk about the AI prediction these days, which is I think it would be complementary, but probably play more and more in parallel going forward to nominate or to identify potential causal variant gene. So the AI prediction is really more from so there's really 2 aspects. One is you really have a lot of literature, gene pathway information already in those model, sort of captured in there, and we just need to get the information out. But also, you can also have a lot of knowledge, learn a lot of knowledge about particular variant in terms of its function. So just give you one example of the latest ones, basically the alpha genome. It's the figure take from their website. So the way they train it, there's 2 key improvement over the previous many models already exist in the last couple of years. One is they take 1 megabase sequence flanking particular variant as an input. So it take basically look at the information not just nearby but also very far away. So they have basically this sort of transformer architecture, encoder and decoder, and then the training that they take all this sort of information, the data available related to the sequence, such as the transcriptome, the splicing, the open chromatin, DNA sensitive site, so on and so forth. So the task is basically if you input this 1 megabase sequence and particular variant, and it can basically predict the gene expression in this area, and also the open chromatin-ness. And then if you put a variant, you see the delta, then you can see whether this variant have that function. So another thing that's important is a really rich single-cell resolution. So basically you can change 1 base and can tell you what's the result. So based on their benchmark, it's quite amazing to see that the performance of the alpha genome is outperform many models beforehand. Even for example the Chrome BP-net is also a deep learning method and looking for open chromatin access. They still even outperform this general model, outperform this specific model. So it's another way to say it is much easier to use this sort of general model to look at all aspects of variant impact and then try to then synthesize the information and go from there. So in here, for example, they can do much better in the splicing prediction, in the QTL type thing, RNA expression, and then the chromatin accessibility as mentioned, also transcription binding site. So the single cell actually will play data I just mentioned will play important role in this. Although the model performance is quite very impressive, but for example if you're looking at the QTL and the AUC curve is only 0.54, it's not that high, right? So it's still a lot of room to improve. It's much better than all the model we have so far. So but still a long way to go in my opinion. So that actually gave us opportunity to basically, use the single cell omics data, particularly in the case of the AMD or the eye, because the training data they use, typically the eye data is not there because the eyes, for many reasons, some reasons a lot of time are missed out from a lot of study, so that's just not available. And then from previous study, it's clearly know the cell context information is important for the model to perform well. So there, if you have single cell multi-omic data, you can do fine tuning. Basically, you take the basic model and you give them the single cell data from the, say, the eye or RP choroid, you'll be able to get a much better model out of it. And also, it's important, I think just as important is the high super functional assay that we can design, and then that can be used to both benchmarking, but also help to fine tune the AI model. But I think between these two, you will get a very good model to perform the task that you specifically need to do over the time. So the future detection, I think still quite, to me, to map from GWAS or other method to get the causal variant and gene is still an open question, and there's a lot of challenge. So I think there's probably many creative ways you can think about to do that. So what we're thinking about is we need to improve. Given we see the single cell eQTL, a lot of QTL are cell type specific. So we think there's a lot of opportunity there. So we need to improve the power of single cell QTL mapping, basically either increase the number of samples and also include disease samples. What I show you is purely unhealthy ones. I know Dr. Morling have a lot of AMD samples, for example. Those would be super useful for improve the single cell eQTL. And then also we need to refine the gene regular elements and link them to the target gene. So histone markers and the ERA and other single cell high C, this type of technology should be applied so that you can make the more solid link between the element to the target gene, because they could be very far away. And also, I think this is very critical is to improve functional validation of candidate variant and regular elements. So I think one of the another bottleneck I see to translate from the causal, from the association to causal is you need to have a very good system to validate your prediction. So we already can make a fair number of prediction, but I think to validate them, that's super difficult right now because you need to find the right cell type. Although this gives you the cell type context, but you still need to have the right system to mimic that particular in vivo cell state and type. And then, and another thing is you need to have endogenous locus modifications. For example, it's CRISPR-Perturb-seq and then gene editing to really looking at the variant function to confirm it. And the last one, as I mentioned, I think there's a great opportunity to combine AI and high super functional assay to make a much better model so that you can more focus your energy on the ones that are likely to be true. So take home message, single cell omics technology from our preliminary data, I think provide a great opportunity to identify distinct cell type and state, and also along with their gene expression and the regular elements and QTL. So that's missed by currently the tissue, the bulk tissue studies. So an integrated single cell multi-omics will enable you to do systematic characterization of the gene regulatory network, connect variant to regular elements to gene in the cell context. And by integrating GWAS with single cell multi-omics, genomics AI, and high super functional assay, I think we will be making a lot of progress toward the causality. So there's a few references I cited, the IGTex study, single cell QTL pilot study we have done, single cell atlas, this in bioarchive, and also database, Alpha Genome. So you can also get access to a lot of human cell atlas data through our website portal, and then if you're interested to explore more, and then yourself. And here's the people I need to thank for the cell atlas project. Thank you very much.
Jason. Hey, Jason Miller, University of Michigan. There's a convincing need for more single cell data. Human tissue is precious, and we have still a lot of bulk RNA-seq available. How effective are deconvolution methods for, in effect, parsing out the bulk RNA-seq data sets into cell types, and then using those to carry out eQTL, type analysis, et cetera? Or are you running into big problems with the deconvolution in the first place?
Yeah. So we actually recently have a publication in Genome Research on that very topic. So the convolution method are pretty good at estimate the cell proportion in the bulk tissue. And then so then if you want to go to eQTL, you can then use that as a cofactor to sort of do the eQTL, mostly to control false positive, to make a new discovery much harder. So it's more to a control false positive issue there. Yeah. Oh,
yes.
go ahead.
Neuvani Milicevic, Tampere University, Finland. Yes, I find it absolutely fascinating that these disease-relevant regulatory motifs are cell-type specific. And then I'm wondering, are they also species-specific? And could that be another challenge in developing animal models and functional assays?
Yeah, I think that's a great question. I think that's one part, the hard part for the validation part, because the coding sequence are much more conserved compared to regular elements, right? So about 50% of element sort of can identify homolog or solog, what I will call you in, say, between human and mouse. But the other 50% from sequence alignment point of view, it's hard to identify. And then if you're talking about particular variant impact on that element, then you have less, be able to conserve that base. That base typically may not be conserved. So that's actually, to me, is one of the hardest aspect. With that said, you do have 40%, right, 50% element are sequence identifiable and probably more functional identifiable. So I think the logic of gene regulation probably much more conserved compared to sequence conservation. So that's people have observed. If you have element, of course, if you have more like organoid or even better, that's be better. But I think still valuable things to do if lack of other in vivo model to put in the mouse or animal model to check the function of the element and also the impact of variant on the element. So still very valuable in my opinion, but it's not perfect.
Hi, Danielle Little from St. Jude Children's Research Hospital. Absolutely lovely talk, Rui. I have two questions. One, you mentioned eRNA on your slides, but I didn't hear a whole lot about it in the talk. Are you going to be using that more in the context of abundance or just as a activity reporter for that enhancer element? And then the second one is, how do you intend to actually integrate this into the clinic? Are you hoping to connect some of these genetic variants and things that you're finding relevance for to imaging modalities or things like that downstream?
Thanks for the question. Yeah, eRNA. So when you go to single cell, if you use the 3' method, you don't get reliable eRNA call. So, but if you use the 5' method, which you do capture nicely eRNA, so most of the data we have generated so far are basically from 3'. So we don't have very reliable pipeline to identify eRNA. But eRNA, I think, to me, is very important because they are active enhancers, presumably, right? So even you don't know which target gene, but at least you know that enhancers, because there's a lot of open chromatin region have this active enhancer histone mark, doesn't mean they are really active in that context. So, but fortunately, we actually from the RPE, starting with RPE, we actually switched to 5'. So now we do have data, we can look at eRNA. So that's a list there. That's like our to-do list. I haven't done it yet, but we're happy to share. The second one, we haven't thought about too much about go to clinic directly yet or the imaging modality, but we're currently just focusing on can we nominate high priority causal gene variant in what cell type, and then can we then validate somehow, then we take to the next level. One thing we do think about to sort of validating is in silico validating is really looking for rare variant enrichment assay, for example, from all of us and the UK Biobank data. But that's still limitation because you only need to go through the ICD code. You don't really know their phenotypes, so on and so forth. But that's sort of validation method, but we haven't seen about imaging yet. Yeah.
Hi there, Roland McGaw here from the University of Edinburgh. Thank you for a great talk. I was just wondering, and I may have missed this, did you have a big enough spread of age of your donors that you can look at chromatin accessibility and gene expression as the healthy retina gets older? Yeah, so we do have a spread from 1 day old to 94-year-old. So it's pretty spread across. There's a lot of genes and also chromatin region that change over aging. But we have not looked into closely other than looking at the pathway. There's inflammatory pathway changes and that, so on and so forth, which sort of expected.
But how do you utilize that for fan mapping? We have not tried it yet.
But you don't have diseased eyes yet.
We only have 4 or 5, but Rob has plenty of it. So. Yeah.
Hello, I'm Gabrielle Rosmek from the University of Michigan. That was a fantastic talk. I'm particularly curious about when you showed the upregulation in one of your specific cell types, the GJA1 for refractive error. Do you know what direction that's going in? Is it hyperopia or myopia, or it's just in general refractive error?
That I cannot recall. We were using that as sort of control. So
Okay.
yeah, we didn't look into closely, but happy if you email, we can show you.
Yeah, and then I just had a second question. For your AMG genes, for that particular upregulation
TGF-beta.
expression, how did you curate that list of AMD genes? Were those genes that you selected, or was this kind of a randomized?
So what we take is we take the SNP, like the tagging SNP from the GWAS loci, and then we take about 1 megabase around it, and then take all the genes and then start doing sort of prioritization. So that's the that's the way we come up with. Yeah.
Kapil Bharati, National Eye Institute at the NIH. Really beautiful talk. I was wondering, you correctly pointed out that to go from EQTLs to gene expression changes, one needs to validate those in functional assays. But I was wondering, using all of your data that you have collected over spread of these samples from very early on to late age patients or samples, could you actually do a secondary level analysis or prediction of what those changes would lead to in terms of consequence of next step gene expression changes in the cell, so as to we could build essentially a digital twin of AMD from early onset changes to all the way late changes, and then start validating all of those in IPSRP or other samples that we have?
That's a very good suggestion. We have not done that. So, but I will think more about it, yes.
Hi, Shayan Ghosh from Johns Hopkins University. Taking cues from the talks in the morning, how, in your experience, do you feel that you need to also correlate with how the environmental factors are affecting these gene signatures, looking into the downstream effect of drug development and particular patient cohorts that we can target?
Yeah, so the current study we do not take into that into account. But I think when you start narrow down the priority gene list, and then you need to bring, or you should bring, the information, for example, the gene pathway, gene function, how that potentially interact with the environment and diet and exercise, so on and so forth. So and I think at that stage it is the right stage where you integrate additional information in. But right now here we just mainly just sort of statistic measurement or functional refinement for gene are likely to be the causal signal for that association study. But downstream you should integrate, yes. Yeah.
Usha Chakrabarti from the UK. So that was an absolutely fascinating talk, and my mind was buzzing with questions, but one question that sticks out particularly is the sort of the racial variation that you get with AMD. And one point that you made was that there was differences in the way of different RPE based on
maybe the transcriptome, yes, yeah.
So, and I also noticed that in your the list of samples that you had, the vast majority were Caucasian eyes, hardly any from the African subcontinent or from Asian eyes, etc. So, what are the chances of picking a gene that is completely irrelevant in this geographical context if you're just concentrating on Caucasian samples?
Yeah, so the sample, we try to be diverse as possible. So about 50% are white by descent. The other 50% are the mix of everybody else, like Asian, Africa, and then the Hispanic, right, how you define it. So because of the availability of the tissue and then the funding, so that's why we will not be able to expand more. So we have not actually dived into say, what do we find? We do find gene expression pathway and chromatin differences between say white and black. So they're there. but we have not dived in because we don't have enough number of samples. We can be confidently say, is this some bias or some biological there. So yeah, I think that's definitely very important actually to move on to expand that study. Yeah. So and the variation of the RPE, so when we first see it, right, the transcriptome, we're saying, oh, is that artifact to cutting them to different pieces? So we actually try to validate it. So I think the most useful validation, there are two. So one is when we get the RPE, we actually did dissection from the same donor, one from macular region, one from peripheral, and then we see how those cells from these two dissections contribute to different clusters, right? That's initially we approximate, are they more peripheral or more central? So we definitely see they go to different clusters disproportionately. So some are more central. The second thing we see there more central is more definitive. Basically, we can derive markers from different clusters, and we perform spatial transcriptome and then hybridization to see how they distributed from AI that we got, whether from white or black. See how do they see distinct localization. We do see that correlate with our findings. So we're pretty sure those are representing different state of the RPE, spatially.
Hi, Rui. David Ortolan from NEI. I actually have a follow-up question on this. Are the markers that you found for the different RP subpopulation somehow associated with, do you see epigenetically different profiles, basically, in the RP population?
Epigenetically, there's very little difference among them. If you look at the chromatin, they're almost identical. Of course, there's DRs, right, but that's really, really small number. Similarly, for gene expression, overall, there are very, very small differences. But there are some genes are higher and lower than the others, But overall, they are very similar.
Martin Paul Agbaga, University of Oklahoma Health Sciences Center. I'm here.
Okay. Here we go.
Really good talk, always moving the field forward. Now, I like your data, especially on the microglia, which validate the role of inflammation in progression of AMD. Now, when you did your chromatin accessibility studies, are you able to pick specific cell types that may at a particular time have more of their complement genes that we had earlier on? How are those genes accessible, and can that be used in future to validate novel findings that you may have in your studies? In other words, if you look at a complement gene, how are those complement accessible? How are they contributing to the inflammatory pool? And can that be used to train your models in artificial intelligence that in future you can identify other pathways that are following that trend?
Yeah, so that's a great question. So, we only did a very cursory, analysis on the data, and then for the complement factor or inflammatory pathway, so the highest actually cell that showed the signal is actually RPE in our data, and then just different depends on different pathway. They have different enrichment and different cell type. I think those data should be more closely looked at. And since we're not an AMD lab per se, so we sort of did a few examples, and then the data is completely open for people who want to explore. And then I think you guys will have more idea than how to explore. Actually, to be honest, than me in terms of AMD. So that's all there. Yes.
Great. Thank you very much. It was a fantastic talk.
Great. Thank you.